So this presentation will be on restriction digests and electrophoresis as a method of DNA analysis. DNA is structured in a double helix with two antiparallel strands. Each strand is made up of monomers called nucleotides, consisting of a 5-carbon deoxyribose sugar, a phosphate group, and a nitrogenous base. The nitrogenous bases interact with complementary bases on the second strand of DNA, forming a number of hydrogen bonds between them, as shown here. This is what gives DNA its double helix structure. Restriction enzymes are typically proteins which digest strands of DNA into smaller fragments. Restriction enzymes read DNA from 5' to 3', locating specific sequences which they recognize and cleaving the phosphodiester bonds between the nucleotides, as shown in the other image. Restriction enzymes originate from bacteria, where they function in defense against viral DNA. The restriction endonucleases recognize and digest viral DNA when it enters the bacterial cell, rendering it useless. DNA fragments obtained from digests can be analyzed using gel electrophoresis, which separates negatively charged DNA based on size. The practical I will focus on is experiment 3 from week 6. As this experiment involves the use of chemicals, safety glasses must be worn at all times along with a lab coat and gloves. When using the electrophoresis apparatus, we must ensure that the leads are connected safely to avoid any electrocution. First, all required substances for the preparation must be obtained, including lambda DNA, restriction buffer, DNA marker, and enzyme stock solutions, which must be kept on ice to preserve the enzymes. 4 microtest tubes then must be labeled with L, P, E, and H for each restriction enzyme and the control, which has no restriction enzyme. Using a new pipette tip for each sample to avoid contamination, reagents must be pipetted into tubes according to this table here. Tubes must then be mixed by gently flicking, and solution must be collected on the bottom of the tube, either by tapping on the benchtop or use of a centrifuge. Tubes must then be incubated at 37 degrees for 30 minutes. After the 30 minutes, 2 microliters of loading dye must be pipetted into each tube and mixed, and then the solution must be collected at the bottom of the tube. Pre-made agarose gel must be prepared by pouring the buffer solution over until covered, and wells must then be loaded with 10 microliters of each sample in individual wells, with the DNA marker being in the leftmost well, followed by L, P, E, then H. The lid must then be placed on the electrophoresis apparatus, ensuring correct orientation of the power leads, and the gel is then run at 100 volts for 30 minutes. After 30 minutes, gel must be removed from the electrophoresis machine and placed in a staining tray to be covered with 120 ml of pre-prepared stain. Staining tray is then agitated for 2 minutes before stain is poured off and the gel is destained. Results are then obtained through observation of gel using a light box. Gel electrophoresis shows how many restriction sites are present in DNA by how many bands appear on the gel. Results of this experiment can be used to approximate the size of DNA strands by using the DNA marker as a standard. DNA markers are strands of DNA with known fragment length. Running DNA markers alongside unknown DNA can allow us to approximate size of DNA strands based on how far they travel through the gel compared to the known fragments. In this image, we can clearly see which enzymes have more restriction sites in the DNA sample, and we can use the marker to estimate the size of each fragment. Apart from DNA analysis, restriction digests can also be used as a process for obtaining desired genes to be used for DNA splicing. By using a restriction enzyme to obtain a desired gene from an organism, and using the same restriction enzyme on the DNA you wish to splice the gene into, the gene can be inserted into the desired DNA strand. Using the same restriction enzyme, identical sticky ends of DNA can be created, allowing the two different strands to bind together. This technique can be used for creating transgenic organisms, which can provide insight into how genes from different species interact together. Gene splicing can also be used to splice a protein-producing gene into bacteria, which allows for large-scale production of a desired protein. Another use for restriction digests is DNA fingerprinting. If we have an unknown sample of DNA, we can compare it to known samples to determine if it matches. Restriction digests are used to cut both DNA samples at specific sites. If any of the known DNA is identical to the unknown sample, then the restriction enzyme will recognize and cut the same sites, resulting in the same number of bands during electrophoresis at the same distances on the gel. This technique can be used in forensic analysis to potentially identify suspects. Thanks for listening.